cup-shaped Demosbong, Maldives. Source G. Warhide. Which came first, the sponges or the comb jellies? The consensus view among taxonomists has long been that the sponges porifera represent the oldest surviving animal phylum. However, recent studies of their genomes have suggested that this title rightly belongs to the comb jellies Ctenophora. The issue has become one of the most hotly debated problems in evolutionary biology, because the answer has profound implications for our understanding of the entire history of animal evolution. Several studies of the question in recent years have come to discordant conclusions. Some have supported the conventional model, which favors the sponges, while others pointed to the comb jellies as being at the root of the animal kingdom. Research is led by Professor Gert Warhide, Chair of Paleontology and Geobiology, LMU, and Professor David Pisani, Bristol University, UK, have now used a refined method to re-examine the datasets employed in these investigations, and their results are unequivocal. The sponges form the earliest branch on the animal's family tree. The new study supports the idea that the results which suggested otherwise were based on the use of inadequate, and hence misleading, analytical methods. All biologists accept that sponges and comb jellies are very ancient groups, which emerged more than 600 million years ago. Sponges are comparatively simple multicellular organisms, which lack clearly defined tissues, such as muscles, and organs like the brain. Comb jellies are structurally much more complex. They use so-called cilia to propel themselves through the oceans, and they possess nerve cells and muscle cells. If Ctenophora were indeed the oldest animal phylum, one would have to assume either that precursors of these organ systems were already present in the common ancestor of all animals but were lost prior to the emergence of the sponges, or that nerves and muscles evolved independently at least twice. Warhide points out that would necessitate a complete revision of our picture of the evolution of the organs and the nervous system. That is the reason why it is so important to correctly define the sequence of animal emergence early in evolution. In order to elucidate the genetic relationships between animal phyla, biologists compared the amino acid sequences of modern species with one another, and construct family trees based on the degree of difference between them. The most likely phylogenomic tree should then faithfully reflect the order in which the different phyla diverged from their common ancestor and from each other. Two of the important datasets used in the modeling studies which identified the comb jellies as the oldest extant animal group were made up of very heterogeneous data. We have now shown that none of the conventionally used statistical methods is capable of adequately analyzing such datasets. Warhide explains. To minimize the compositional heterogeneity of the data, the authors of the new study employed a procedure in which the amino acid sequence data were first divided into groups based on the biochemical functions of the proteins they form, and then subjected to the modeling process. The analysis of these datasets, including those derived from the data that had previously ranked the comb jellies as the older phylum, demonstrated with high statistical confidence that the sponges are in fact the oldest of the animal groups. Our results confirm the classical assumptions concerning early animal evolution, and should help to put an end to the recent controversy over the origin of multicellular animals. Warhide concludes.